This is where we're at. So you're incorporating this into your capstone journal. And that can be direct thoughts about what you're thinking about your project, and those thoughts can move around. And then they also could include thoughts from this class. So what it's like, one way to think about your capstone project is you're going to pursue one of these topics. You're going to pursue a topic in great detail. And how to get to all the details of the science is to be participating in all the other topics we talk about. So in every example we give, it's an example that, oh, that could be a capstone project. Or it could help illuminate the science behind the capstone project. In class, we're not going to really talk too much about the other pieces of what are necessary to put down the capstone project together. But for example, today on Brightspace, I'll post an announcement that describes what we need to do for our first writing assignment, and it's like a 2-page writing assignment, like that. And so there are pieces that you're going to be working on in your capstone project that we won't discuss a lot in class. What we will be doing is discussing the science of it, and the science includes that sort of term paper concept of you're going to try to be collecting research on this topic. And part of it is that we want to stay focused on a topic, but we want to appreciate the depth of range of aspects of science that relate to that topic. So we'll try to talk about that way in class, and you'll see we start talking about that way today. So then we're talking about chapter 2. We do not have class on Monday, so next Wednesday we're going to do a discussion on chapter 3, that's on Closed Reserve, chapter 3 of the Gilbert book, and that's about symbiosis, especially focused on plants and animals, symbiosis with bacteria, like that. And so gut microbiome and things like that. And so we're going to talk about that some today, but probably not enough. I'll try to get to it, but I want to follow up on some of the mechanism stuff and dive into that a little bit more, okay? And then we'll have that writing assignment we'll do in a couple of weeks, okay? Does anybody have any questions about what's going on or anything at all? So let's just talk some more about mechanisms, and we'll probably talk mostly about mechanisms today because that's relevant for what we talk about in the symbiosis. So, you know, I'm supposed to be setting the stage in the lecture, but I think I should spend lots of today kind of help framing what do we mean, you know, when we dive into the topic of symbiosis with regard to mechanisms. And so we set the stage, and there's a couple of kind of different angles that we want to work with. So here's the first thing. So this guy, Conrad Waddington, a geneticist in the 1940s, he coined the term epigenetics, and it really means just changing gene expression, that we have more DNA than we're using, and so we can change it. And it can be changed in a variety of different ways, but of course it can be changed, in our interest is changed based on the environment. And he created this drawing. And the drawing is like this, where the ball is starting at the genotype, and the ball is going to roll down and produce the phenotype. And the surface is a kind of an odd combination of both the environment, but also the potential the genotype has to get to the phenotype. And so he was, you know, this is back when genetics was theoretical. They didn't know what a gene was. They didn't know that, you know, how it was organized in DNA. And so, but you get the idea of this plasticity in the ball. So let's give an example of that. So we talked about that bacteria. And of course, we're not spending much time talking about bacteria this semester. But if the environment includes lactose, then the ball, the genotype, will roll down to the bacteria that makes the enzymes, that has the enzymes that can degrade and use lactose. On the other hand, if lactose is absent, it can roll down this. And so the surface is both the idea that there's an environment with features, and then the capacity of the genotype to roll down those paths. So it's a kind of a combination valley thing that's going on, right, like that, okay? So let's, when this is out of place. Well, let's, since I might not have put it in another place, so let's just talk about these terms also here. So plasticity is flexibility in the development, okay? So these are terms that he made up that would be the opposite of plasticity. And the most common term that people, and these people use these today, and they say canalization, which comes from canal or valley. And canalization means that you don't have options, that you're just going to roll down and make that thing. And that would be the direct connection between, you know, red flower crossed with a white flower. Oh, we get a pink flower. And it doesn't have anything to do with the environment, it's just going to roll down that way. So he used this term canalization, and it's good to think about that, you know, just to think about how the genotype could create these huge, deep valleys, and it's only one way down, like that, you know? A good example of that would be, and I know some of these examples sound kind of odd, but human body temperature is really canalized. You know, it's almost always 98.6. And that's because we're warm-blooded, and it's not sensitive to the environment, right? Some other organisms are cold-blooded. Their temperature is totally sensitive to the environment. So it seems like it's very canalized. That's a good example because, you know, most of the time you are. But you can actually get a temperature when you have a fever. So there can be an environmental factor that changes your body temperature. It's just unusual, but you can see how it's a selective advantage to have that. In other cases, you know, maybe there would be absolutely none. And so that's called canalization. Other biologists today would call it, it's very robust. And that just means that what you start with genotype is what you're going to get phenotype. And so that would be examples where no, there's no plasticity. It's canalized, it's rolling down to a very specific phenotype. And that phenotype is very robust. You know, different environments, we don't care, it's going to go to that, right? And here's a very robust phenotype in us, 2 eyes, right, like that, really locked in on 2 eyes. Some species, you know, probably make multiple eyes, you know, like that, but we're really locked in on 2 eyes. And so there are lots of examples of that, but it provides kind of the idea of, you know, well, is it really highly canalized and robust, or is it plastic, like that, okay? All right, so then we can go to our example here we talked about last time, right? That's chapter 1 concept, and we're talking about the mechanism and stuff like that. And to point out, you know, we dive into the mechanisms. It's in that table in chapter 2. Those are the beetles, horn growth and horn size, and they're really affected by these 2 hormones, ecdystron and JH. So, you know, we want to be diving into those molecular mechanisms and also diving into the biology of the horn and the horn size, okay? So in all arthropods, it's juvenile hormone that modulates, well, I didn't include that, juvenile hormone modulates this activity, okay? So we can manipulate in labs, experimentally, the presence of that horn or absence of horn because we know mechanisms. So that's an important concept, and there's a lot of details about that. And I have a slide in here somewhere. Where did it go? So here's that slide where you say, okay, but does this example fit the model of epigenetics? And it does. And so here the idea is the environment is that surface that the genotype is rolling down can either be low nutrients or high nutrients. If you're going to be low nutrients, you're going to make a small adult male beetle, and it's not going to have a horn. So it's making a choice. And this gate you can think of on a surface like that. So there's valleys down this way, and can it go down this valley or that valley to the final phenotype? So that's the idea. And I should have had this before those slides about the details of the mechanism, you know, because it's a gate like that. And we talked some about that last time, about how it's it should be a polyphenism. It should be a gate like yes or no, right? Not just a smooth, wide opportunity, like a really smooth, wide valley where you could be anywhere along the width of that valley and be, a very plastic reaction, okay, like that. So a lot known about this. The person who's going to pick that topic is going to dive into that, and really almost all the topics that look about insects really dive into the hormones and how they regulate hormones like that, right? And many of you will have to deal with hormones in your system, because here's a different figure from the second chapter, and it's actually a different nuclear receptor called the glucocorticoid receptor. And we brought that up a little bit previously, where when there's a chronic stress in the environment, any vertebrate can respond in a stress response that's different than the adrenaline epinephrine response that is a very short-term response. And so, you have to dive into hormones and like corticosteroids and nuclear receptors like that. So lots of times, we'll end up talking about nuclear receptors in class and in your topics, okay? And so that idea. And so here are these beetles, right? And so now I just want to talk a little bit more about the mechanism of the story in those beetles, because there's something we haven't brought up yet about them. So we talked about, it turns out it's that ecdysteroid. So it fits into the juvenile hormone and ecdysteroid thing. And the insect often do fit into that, okay? So here's another aspect of this that is something that the person who would use this kind of topic had to dive into. And so the idea is, we didn't get to this yet, and the idea is that there are both great details of molecular mechanisms, and then there are details of our understanding of the organism and organismal concepts, and we have to pursue both of them. As for example, at the end of the class on Monday, Ashton said that very interested in immunology and immune system thing work. And yeah, that'll be good, really like that. And that tends to lean towards cellular and molecular immunology, but also have to deal with how this fits into the organism's life. And that's really challenging. And it might include looking at other species and how their immune systems work and stuff like that to be informed. So here's just an example of that here. So if the difference is the adult male beetle, and the smaller ones have no horns and the bigger ones have horns, okay? So this is just a graph we talked a little bit about. In general, there's a correlation between animal body mass and body parts, like that. I'm a big person, I have long arms, like that. And it's partly genetic, partly I just chugged milk all through my childhood. And that general correlation is broken here. And we see the polyphenism, right, like that, in the horns, okay? All right, so there's two terms here, and that is, generally there's truth in this scaling of body size to body part size. And so a good example of that is our heart. So bigger people tend to have just physically bigger hearts, and it's pretty well correlated. And that's got a term, and this is where we dive into this organismal level biology a little bit, just to see that you have to deal with it, those facts too. That's called isometric growth in body plans. And that means the ratio of a part is correlated with the full size, like that. And there's a lot of that. With the beetles, though, right, we're not talking about that. So if we looked at the horned beetles, there would be an isometric relationship between their horn size and their mass. Because the big beetles range in size, and their horns range in size, and the horned beetles tend to have an isometric relationship between body mass and horn length, like that. An alternative to isometric growth is called allometric growth, and that just means the ratios are not scaling. They don't match, and that's really common, and there are lots of examples of that in our body and stuff like that, like the size of our brain. When we're a newborn, we already have a really big brain in a little body, right? So body size to brain size is allometric in us. So this is this odd area of biology. I don't know if it comes up in any courses, in undergrad biology courses, but it's a huge field, and it's broadly called allometry. So one of the features is either they don't scale, it's allometric growth, or they do scale, it's isometric growth. And so allometry is the study of that, and somebody that wanted to dive into examples of these body shape sizes and things like that would need to dive into this field, should dive into this field of allometry. And it becomes a big story about math. and relationships in math. So isometric scaling happens when proportional relationships are reserved in size changes during growth or evolution, like that. And so that's kind of the default, is probably these are going to scale together. And if they don't, we're interested in why not and how that works, right? So that's allometric scaling. It's any change that deviates from this isometry, like that, okay? And so that concept's been around a long time. And in this field of allometry is this idea of the sizes of species, and that actually comes up in a lot of examples in our course, is this that ends up being the size range of the species, and how that works. And so you start out with some of the topics in our course. A person, even a scientist, a biologist, would still approach kind of like somebody in the general public would be like. They'd just be, yeah, that's a fact of life, There's a size range of this particular species. Mice are smaller than humans. Both have a size range, but they don't overlap at all. Like that. But it turns out there's a lot of biology there because it's a very significant evolutionary heritable trait, these features, and they are evolvable. So, really need to. And so, like, Ashton's really interested in the immune system, but he needs to try to wrangle with the role of the immune system in the organism as well, and how that fits into the evolutionary and the niche and the story of that species, like that, right? Okay? So just kind of rolling through this to kind of add that to that mechanism stuff, is that in these topics, there'll be mechanisms down at the molecular development level. We really want to get to those, but also there are these not just stories, but biological research at the organismal level and how that fits in. And it might be the organism roaming around in its immediate environment. It might be the life cycle in the organism and how that fits in. Like, how does that nematode that can extend its lifespan, how does it fit into the environment, in its natural environment? So we got to deal with those, because it's both ecological developmental biology, like that. And so there are examples here like this, with the social insects, and just to add a little bit to the molecular mechanisms of that, we said the reason the queen bee or queen ant or queen wasp gets formed is because the workers feed the larva this royal jelly. And so the royal jelly actually is just material nutrients that cause the increase in juvenile hormone, like that. So just to talk about that and follow up on some of the things that came up. So then, juvenile hormone and DNA methylation are interconnected, and high levels in the larvae induced by queen destined diet, royal jelly, are associated with lower DNA methylation compared to the workers, like that. So we want to dive into molecular mechanisms and tie them together, tie those molecular mechanisms of development into the overall body, and or story example, and then fit that in, like that. Like somebody who would be pursuing this would be like, well, what in the environment causes those workers to do that? Because they're actually creating this royal jelly material, and they're doing it by adding different nutrients to it. So what's causing them, right? And so we have to also address that aspect of the environment, it's going to be the queen, and the queen's got this royal jelly, but, we can't just dive in and say, oh, juvenile hormone is destroyed, and, molecular mechanisms, stuff like that. We also stay up at the level of the ecosystem, the ecology, the organism, and how it fits in, like that. Like, why do they need a queen? What are the cues that they would be producing a queen, right? So we have to scale it up to that. So it might include, for example, in chapter 3, the relationship between the molecular mechanisms in both the host and also those microbes, and because there are two stories going on there that we need to deal with, like that. And so just kind of trying to zoom into that a little bit and stuff like that. Okay, so then continue on that allometry in the examples in chapter 2 and 1 that we're trying to get to. There's a lot of them that include this story concept of allometry. So the scaling is not just a relationship between mass and other odd body part, but also things like scaling between mass and physiological functions like metabolic rate and different topics like that. There's also allometric or isometric scaling between those. Yep. Okay. And so one of those is the muscles. And so we talked a little bit about muscles, and that comes up in our course. And there's a scaling relationship between muscle size and the organism. Here's an example that came up in chapter 1 of, we talked a little bit about these tadpoles, and different species of frog have tadpoles that grow differently depending on the environment. The most common thing is the tadpole can go through metamorphosis quickly and early, like that. Here's an example where if the little tadpole environment is really crowded, the tadpole can grow up with a different shape, and it has a different shape of mouth, a different digestive system. And if it's crowded, this species of tadpole grows up to be a carnivorous tadpole, and it eats its brothers and sisters. And that sounds really contrary to natural selection, right? To eat your siblings. But the idea is some of us are going to get out of here. Mom screwed up, and she didn't get enough food at the grocery store. So, well, we're just going to start eating each other. So some of us can get out of this situation like that. And in that example, there's scaling things going on here, right? This guy's wider mouth, bigger muscles, jaw muscles, different. So that also includes that science of these scaling relationships, and how is growth regulated? How is it that those muscles grow bigger? What are the cellular mechanisms of that? But then putting them back into the context of explaining and diving into the organismal level research in understanding bodies and body relationships like that, right? So here's an example from your book in chapter 2 where it's talking about muscles, and this example is talking about how in our bodies, and we just kind of take it for granted. So some of this stuff is, you take it for granted and like, but our muscles can get bigger depending on our activity. So somebody can, lift weights and their muscles can get bigger. And there's a lot of molecular mechanism understood about how that happens. And there's some stem cells that get activated by mechanical strain, right? The student can write about that. But also needs to write about how that fits in. Like, why do you suppose our species does that? Because it appears that other mammalian species don't necessarily do that. Like if you get them to do the different kind of activities, that some of them don't get bigger muscles. Their muscles change some, but it's not that they're getting this muscle hypertrophy like humans can. And then what would it fit in? What would be the evolutionary story of that? What's the history of the species that would lead to that plasticity, right? Because remember, if you don't use it, you lose it, right? If you don't use the plasticity, you'll lose it, and it'll get really canalized, right? You won't have that flexibility. So it has to be something, and so we got to wrangle at those levels with mechanisms. Down into the molecular details, and for some topics that's a big challenge because is there not much known there and things like that. And I'll give you an example of that. One of the examples in our book in chapter 1 is how the jaw, well, we'll just use that same tadpole one, but one of the examples in our book is about a fish and how the fish grows a different jaw, a different mouth shape depending on its environment. And it turns out there's a lot known about these vertebrates and how neural crest cells form their face. So, the frog tadpole has a different face, so the stem cells have to be making a response. The fish has a different face response like that. And the fish is just a species that nobody knows too much about. They just know at that level, and you probably write and understand how it fits into the environment given its face and ecology it fits into. But what about the diving into micromechanisms? And sometimes it needs this, where it turns out that there's a very, very well understood vertebrate model organism called a zebrafish that's studied in lab. And so anybody that would be studying a different kind of fish, the environment, they would probably need to learn about the mechanisms using a different species like zebrafish, right? Like that. So that's the case. And, you know, that would be true in humans, right? Is that, well, let's look and compare it to other species of mammals and vertebrates like that, and what's known about the molecular mechanism. So there's a lot to do with regard to diving into the science, and that's the kind of the take-home message on this, okay? And because here's some molecular mechanisms. Here our book is talking in chapter 2 about these molecular mechanisms. And it includes things like Tor, you know, and we ran into Tor before, so Tor comes up frequently like that, okay? And so then back to these beetles. Oh, why did I put this way? Oh, yes, so back to the beetles. So here's another thing that I that I just briefly mentioned before and wanted to talk more about in the mechanism story, and that is the nervous system. So in these beetles, these are not learned behaviors, they're inherited behaviors, they're instinctive behaviors, and the behaviors must be based on how the brain is formed and how it gets wired together in the development of the brain. Because it's sort of a robotic behavior, and so they must have different brains. One way to think about it is that, you know, it would be interesting, like right in us, we would be like, well, I could just shift from one or the other depending on the need. My behavior is very plastic, a lot of learned behavior, a lot of plasticity just in how I can do my behavior. But these guys can't, you know, like that. You know, they can't shift into the different behaviors. So there must be an evolution of their behavior. It's based on the development of their brains, like that. And anytime we talk about behavior, that's going to be the case, right? So here's an example of that too. We said, oh, you know, in this case, if you remove the male, the largest female becomes a male. And we can dive into the development of the reproductive system and stuff like that. But also, what about the behavior? And in this case, how can the behavior be plastic like that? What's going on like that? And so we talked some about hormones and how hormones can regulate behavior and other, you know, development and other traits like that. And so I just wanted to talk a little bit more about the nervous system today. Woo! And so I just want to talk about neurosecretory cells because they are very often regulating hormones, and so here I've just, you know, did an AI overview to say, you know, neurosecretory cells, do they affect hormones? Yes, they affect hormones. And so I just want to describe the nervous system, and, you know, this is a course where, well, you weren't required as a prerequisite to take the neurobiology, but for some of the topics, if behavior comes up, either it could be hardwired behavior like in those beetles, or it could be plastic behavior, right, where the fish's brain has the capacity to behave one way or the other. In both cases, what are the mechanisms? And so here, you know, we're talking about molecular mechanisms, but also cellular mechanisms, right? So we have to be ready to talk about the organism in its environment, the organism in its natural history and evolution, and also the molecules and molecular pathways that regulate the development inside the animal, and also the cells and the role of the different cells like that. So this is just a little bit about cell biology, and it goes like this. So and part of this message is that when we start looking at the development and physiology of systems in animals on Earth, lots of times they're astonishingly similar. And that's really true about the nervous systems on Earth at the cellular and molecular level. So we see a lot of different nervous systems, but they're all really similar in their organization like that. And so we could say, like life on Earth, in a nervous system, there are really just two types of cells. There are neurons, and there are glia. And neurons are the cells that are going to guide any behavior, and glia are helper cells, help in a variety of ways. So we'll focus in on these neurons, and we can draw a picture of a neuron, and a neuron has a cell body, and the cell body is where it's the biggest part of the cell, and it has the nucleus, and it has this most of the cytoplasm of the cell, and coming out of the cell body are these two growth processes like this. They're very thin. I drew them kind of wide, but they have cell membrane around them, and they're like that. In some cases, just to show you this, in some cases, like invertebrates, a lot of times the cell looks like this, where the cell body has one kind of outgrowth that just pokes out a little bit, and then the processes go like that. But in us, it looks like this, where there are these processes, and sometimes the processes can have, there could be multiple processes coming out of the cell, or they can be branched like that. But they're fundamentally two, and that's true for every neuron in every nervous system, I'd say. And one of the processes, or group of processes, is called dendrites, and they're going to be bringing essentially the communication electrical pulse in toward the cell body. And then the other processes are called axons, and they're going to be bringing the information out away from the cell. And at the end of both processes are a synapse, and that's where the cell body gets, the cell process gets really close to another cell. So these are both synapses. And what it's like is the message will go from across one synapse to the next cell, and then continue this essentially an electrical signal. An action potential is what it's called. Okay. All right. So that's the structure of them, and then there are really just four kinds of neurons on our planet. Given their structures and the four kinds of neurons, and we'll do this in a human. And that's true for, I will just say, vertebrate. So vertebrates look like this, and they fit in this way. And there are some differences in invertebrates. But we'd start out with muscle neuron. And all the muscle neurons in our body that cause muscle contractions are inside the central nervous system. That's made up of the brain and the spinal cord. And so what we mean by their inside is the motor neuron has its cell body in the nervous system. And then, in this case, right, it's going to have dendrites and an axon. In this case, the axon is going to leave the central nervous system and go out and make a synapse on a muscle. And cause the muscles to contract. And so that's a great name, motor neuron, because it's our motor that moves stuff around, or moves our arms and legs around like that. And so in this case, what we have is the cell body is in the central nervous system, and as it develops, it grows this axon out to reach a target and make that connection. And that would be an axon, the information is going to go out. For this motor neuron, the dendrite is going to be connected through a synapse to another neuron. And that neuron is going to cause, a signal that will lead to muscle contraction. So I'm just going to redraw this now to emphasize the other neurons. So if we just go like this, and this is the central nervous system. And now we're just going to draw the motor neuron really small like that. There's the motor neuron. And there's the axon coming out, going out to synapse with the muscle. So it's going to have some dendrites, and what we'll do is we'll draw dendrites like this. So there's a dendrite there, and here's another dendrite. So they'll make connection with the two other kinds of neurons that are inside the central nervous system. Okay, so the second kind of neuron we're going to draw makes connections like this, and I'll make it pretty small, and it's called an interneuron. Oops, sorry. And interneurons are what we do when we're thinking. So that's what is inside the central nervous system and is processing things. And the entire interneuron is inside the central nervous system. So its axon is going to grow out and make connections either with other interneurons, right, lots of fancy thoughts, or directly with motor neurons. And then its dendrites are going to be bringing signals in. And the signals could be coming in from other neurons, interneurons rather, or it can be coming in from a third kind of cell. And so we'll make a third kind of cell, and we'll have it connected here. This kind of cell also has its cell body could be here, or more likely its cell body is near the central nervous system in a region like that. So we could have a cell body for this next kind of neuron that might be inside the nervous system or nearby, like that. See either kind of location like that. And this is a sensory neuron. And its axons are all inside the central nervous system. So its axons are going to be sending information there. And then its dendrites are outside the central nervous system. And they could be, receiving sense of touch or heat or pain or just where our bodies are in space, and that would be where their dendrites are. And from this, you can see simple behaviors like reflex behavior. And a reflex behavior, like when you kick your knee when it gets hit with a hammer, just involves a circuit that's a sensory neuron connected to a motor neuron, and they just click like that. Much more commonly, the sensory neuron brings this information in, and then it's processed by a whole bunch of interneurons that then make a decision, like that. And you'd be like, oh, our nervous system seems more complicated than that. It is, because there's a lot of interneurons. And then there's this fourth kind of cell, and it's the one that comes up most often in our course. And it's just interesting to see, we're at the level of the neurons, and maybe there's an exception to this, but I'm pretty sure there are only 4 kinds of neurons on our planet, like that. And they would be motor neurons for output, they would be interneurons for processing and thinking, and there would be sensor neurons for input, like that. And the fourth is called the neurosecretory cell. And neurosecretory cells are output. And I should have drawn this down here just to include it from the other figure that I drew. And that is, there's a muscle. Motor neurons bind to muscles and get muscles to contract. And so the other kind of neuron is a motor neuron. And I'll just draw it down here. They could be, their cell body could be in the central nervous system and in some other locations. So this is a neurosecretory cell. And neurosecretory cells are neurons, and they're not interneuron, sensory neuron, or motor neurons. And what it's like is their dendrites will make connections with these other types of neurons. So they'll have dendrites for input, And then their axon goes out, and since it's vertebrate, we'll depict it like this. They make, it's not exactly a synapse, but we could call it almost a synapse. And typically they make it on something like a blood vessel. And these other neurons, when they want to communicate, so the sensory neuron, the muscle neuron, the interneuron, when they want to communicate, what they do is at that synapse, those release neurotransmitters. The neurosecretory cell isn't a synapse because it doesn't release neurotransmitters, but it releases molecules in the same way, and those molecules are hormones. So we talked before about how important the connection is between the nervous system and the endocrine system, and refer to it as the neuroendocrine system, and neurosecretory cells are the way that gets done. And so it's really interesting to see that, because you can see how connected, how intimately connected the nervous system is with the endocrine system. Because even though lots of hormones are not made by the neurosecretory cells, for example, estrogen is made in the ovaries and testosterone is made in the testes, like that. There are a lot of hormones that are made not from neurosecretory cells, but a lot of times neurosecretory cells make a hormone that controls them, like that, okay? So what's a good example of that? Yeah?
Yeah, I was just gonna ask, like, we talked about last time, the RAS, how it, like, visually sees, like, other, like, that it might, like, female that it might be larger, something like that, like, fit
Yeah.
into.
Yeah, right. And so, you're used to thinking, like, when I see stuff, I can respond to that, like, I can walk toward, that, chocolate shake and drink it, like that, right? So when we see stuff, we can also regulate our neurosecretory cells to change like that, yeah? And regulate things like that. And there's some famous tissues that have a lot of neurosecretory cells in them, like the pituitary gland. And, okay? So then let's just talk about, just to plug this into some of the examples. So with these, let's see, what's the best example we could think of? Well, yeah, the insect ones that what's being monitored here is the nutrient status, and that's not such a great example to think about how our nervous system can contribute to regulating or responding to nutrient status, because you'd think it's just more at a molecular level. So those aren't great examples. I'm trying to think of an insect example. I guess I can't think of one right now. Yeah. That would be real satisfying, where you'd be like, oh, that would be like these. In the vertebrates, the neurocirculatory pathways are more complicated than in an insect. And so that's why I was trying hard to think of an insect example to kind of keep it nice and understandable. But I'll think of an example, as we go ahead, and help you connect that, connect the nervous system's sensory input with regulation of neurosecretory output, okay? And so, just a little bit of information, right? And, you can't use AI stuff to write your paper, but this is just the first example of like AI can be used super helpful, right? To just get an overview, and it's got a link, and there's some sources, so you can totally use that. You just have to include that as a source. I'm using the, got this from the AI overview in your journal, like that, absolutely fine. we're not going to use it to write our paper or chunks of our paper, but it's super helpful, and it's important for us to, you know, use it in our research and our fast learning, right? We're like, oh, okay. So I'll use this a lot, where I just, you know, now when you Google it, if you put it on AI mode, it will give you a summary instead of just links to research articles. And so just to show you a little bit more about that, right? It gets us a good introduction to that. And then here it's describing, you know, here a description of how, you know, we make the connection through these neurons from sensory input to motor output, like that. And you know, just examples of neurosecretory cells in vertebrates, and again it gets complicated, regulating other hormones, because that's very, very common, like that. The pituitary gland will regulate the hypothalamus and so forth, and then go on to regulate like the testes, okay? And this is a little bit different topic, yep, and yeah, we'll get back to that, yep. And so then here's this neuroendocrine axis that we want to be aware of when we're talking about behavior. We really want to dive into that. And my point of talking about in that some detail is it's more at the level of cells and organ functioning, like that. And in our course, in our topics, we want to range across that, you know, from the ecosystem and evolutionary history and natural selection to the organism and what's going on in the organism. Is it a question of allometry or what is going on that we're studying, down into levels of organs and levels of cells, and cells like how neurons function, and down to molecular mechanisms of gene expression and enzyme activity, like that, yeah? And for just about every example, for every specific capstone project, you'll find the science to seem like it's most accessible at one of those levels, or two, you know, like that, where you're like, I'm gonna get down to the level of that hormone, it's a nuclear receptor, I'm just gonna write about that a lot. And you can do that, and you gotta do that, you know, to dive into that, especially the, you know, the level where there seems to be the most science focus, and we're gonna really write about it, and, you know, add to that. But also, we gotta challenge you to incorporate understanding and the scientific understanding at these other levels, like that. And, you know, and I'm a good example, right? We talked last time, you know, where I said, you know, anything that's not at a molecular level is stupid, like that, right? And you probably have those biases. The reason it came up because Ashton confessed that that's his bias, like that, you know, I'm just like, okay, you know. But it's gonna be challenging, right? Because like the immune system has a lot of cellular, a lot of molecular mechanisms, stuff like that, but also, how does this fit in? And I gave an example of how it's really valuable at a molecular level to appreciate those higher levels of organization. And I gave that example in my own research of how the species I study undergoes overwintering. And that's fascinating, and it led me to, you know, research that led to a couple of really neat publications and a research grant. Just being, you know, mindful of that. So we're trying to do that in our work, like that. And so that's some stuff like that, and I have more of that kind of stuff. And but at some point also today, I just wanted to pause and talk around in the class about like how you feel. Just, you know, you could just say, I'm just getting started, like that. But like when you're thinking about your capstone project, do you have some thoughts? Because it is also helpful for us to, you know, share. And so let's just take a few minutes and we'll just go around in a circle and say, you know, what kind of things you're kind of thinking about. They could change, and you might be just like, yeah, no, I've been busy with other stuff and I'm just getting going. But Ashton already mentioned something, and so can you just kind of loop through that again and say what you're especially or might be interested in?
I'm gonna write about the plasticity of immune cells.
In vertebrates.
Yeah.
Yeah, and so that's a good example. There's a lot of ongoing development in the immune system. That's a big strategy in vertebrates to defend themselves from foreign infections and stuff like that, is to just have a constantly renewed immune system. So we've got our bone marrow cells constantly making new blood cells, and constantly making them fresh like that. And so there are lots and lots of that, and so that's a good example because a person could just get completely blown away by all the things the immune system does, and we need to kind of limit it and focus in on certain features of it. One of them could be acquired immunity and memory cells. So when an antibody producing cell gets stimulated, it will turn into a memory cell, and that's how we have our lifelong immunity to certain things like that. And that's a kind of plasticity that's going on like that. Yep. To talk about that also, one thing we should talk about is, we've been talking about the cell, and it turns out that the cell, of course, is the fundamental unit of life. And for the cell, everything is the external environment. So we like to talk about, the external environment being outside of us, like that. But like say with the immune system and in any system, it's really what's going on outside the cell. So there's an internal environment, the hormones, for example, like that, right? Internal environment that also is relevant, and the signals that are coming from that internal environment. And that's not, like tricking the system. It's just realizing that when we think of the environment, and we usually think of the external ecosystem, but we can spend time thinking about what the internal environment is and how that's a factor. Like that. Yeah. And then, here's something that Ashton's really interested in. Have you taken immunology as a course? Yeah. And so, can build on that. And that, that's great. But it's, we'll have different examples like that. Juliet, are you thinking about a topic or anything like that?
I've really just been collecting potential topics that the textbook brings up.
Yep.
I thought toxoplasmosis it brings up, which I thought was really interesting, and how that relationship can be evolved, because it specifically goes after the rats because they know it will be eaten by the cats, which I thought was cool. So I think maybe looking into that.
Yeah, and so that would be this idea of this symbiotic relationship and how that evolves and how there is this plasticity and how we look at I mentioned before, a lot of times parasites or infectious organisms can have really complex life cycles, and that's based on the different environments. So some of them require like multiple hosts to go through their full life cycle and stuff like that. And then there can be plasticity in that, and so we also want to, in all these projects there'll be this thing that's a challenge, where you're like, oh, this makes sense. But then at what level is it a challenge, and can we push into that and write about it, but also find research topics? And that's the main reason why I wanted to spend time talking about allometry, because there's a good chance that none of you have heard of it, but yet it's, oh yeah, duh, okay, that's fine. But it's actually a big field of study. It has been since forever, and it becomes very interesting, the concepts and principles of allometry and that scaling. Yeah. So that sounds great, and but if it changes, that sounds great too. Yeah. And Mike, are you thinking about something?
Yep, I'm gonna do something with the behavioral plasticity of cephalopods.
Okay. So that's like an octopus.
Yeah, I'm probably gonna do the octopus because they have the most studies on them. I want to do squid because I like squid more, but there's very few studies on squid. They're mostly on octopus, so it'll probably be centered on octopus.
Yeah, and so that's a thing, right, where a person can pick an organism, and there's a lot to say about octopuses, and but sometimes you have to look at some other cephalopods to try to really drill into the science understanding, stuff like that. Yep. And do you have a sort of topic that you're thinking about?
So most of the studies so far that I've looked at are based on introducing novel predators into environments that they don't exist in, and then watching what the octopus do in response. So it'll be something that they've never been around, and then they'll figure out what that species is doing in the environment. environment and then either mimic it or make a new defense system for themselves to get away from it. And it'll be something that no other octopus has ever been seen doing. So, yeah, something around that. It'll probably be based on predator introduction, mainly because that's so far,
Yeah.
all the studies are on that.
Yeah, that sounds good. You know, I think that project, you know, is the thing where, like, it's great, really great stuff. But I'll always be trying to push it toward developmental stories, more so than, physiological stories. And sometimes, behavior modulation is, like that plasticity, I would consider, kind of primarily like physiological, because it's like, oh, it's got that. The plasticity is in kind of the physiology rather than development, because it's ready to, it's got a flexible brain. And we can talk about that, but yeah. And I have to think about this too, like I'm not, like one of the reasons I'm a molecular biologist is because I really like nuts and bolts stuff. And I really, I don't spend a lot of time ranging about my thoughts across these scale, from like, oh, this is going to be about, adjustment of, novel behaviors. Yeah, but yeah, and we definitely, all of you, I've talked to a couple of you, a few times already about ideas and thoughts and kind of brainstorming it, and we'll want to do that with each student, and we won't be having time enough to do that in class. We can, inject some in. Bryce, what are your thoughts so far?
I've pretty much just been reading and trying to wrap my head around the methionine restriction in the MEST protein. It's kind of
The what?
in the MEST protein.
Oh, yeah, you and I talked about that, but
Yeah.
nobody else knows what that is.
Yeah, no, I honestly, it's been like kind of difficult to read some of the papers on Rob Koz's website. It's or on the website, but his articles. It's
Yeah.
Just because like it's very like nuts and bolts stuff. So I'm just I've just been trying to wrap my head around it and see if it's going to be something that I think I can address and like, be simple enough. You know what I mean?
Yeah, well, yeah, and so MEST is a protein in mice, and I mean, there's probably a human ortholog of it too. And just to quickly sort of describe, I talked to Bryce about what he was interested in. I described there's a researcher at Maine Health Institute for Research down the road here in Scarborough named Rob Koza, and he studies this kind of topic, and here's a specific experimental experiment they do. So they'll take mice, laboratory mice, and there are a variety of different strains, it's like breeds of mice, and some breeds they're interested in because they have this effect more dramatically, but all of them, have this some. And one experiment goes like this, where you just watch a lab mouse and it'll eat a certain amount of the lab chow food, and then you can just leave excess food and they won't eat it all. And they'll just grow up to be a nice, healthy mouse. And so you can take that food and without having them eat more, you can just increase the calories in the food they're eating. And they grow really obese. It's the same mouse, it's the same basic nutrients, and you just had it's high calorie, we call it high calorie. And so you have, two groups of the mice like that, and they're the same breed, so they're, doing the same thing. And you give them this high calorie diet, and they get really obese. And one group, you just keep feeding them that, and they get, more and more obese. That's not changing. But the other group, you give them the same high calorie diet, you just change one thing. And the one thing you change is you decrease the amino acid methionine almost to zero. It is an essential amino acid, so you got to have it in their diet, but you can give them just a tiny amount. And it's difficult to make that kind of diet because it's, just in protein. So you're not giving a protein diet, you're giving kind of an amino acid diet where it has all the amino acids in both these and they're doing fine, but you just reduce this methionine. And so they call it a methionine restricted diet. And it's got just as many calories, but just with that one change, in like 2 weeks, these mice are all normal weight. They were obese and they were heading with this high calorie diet to just become more and more obese. and they've just gone down to a normal mouse's weight. They're still eating all those calories, but they're dealing with them differently, like that. And so his lab researches that. It's nutrients, right, environmental factors in this plasticity, the developmental plasticity of growth and obesity, and how dealing with those calories, right? And it must have something to do with how the organism is monitoring its diet, not by looking at the overall quality of the diet, but using certain like hallmark molecules to use as a thing, as a measure of what kind of diet this is. And the mouse is just going like, whoa, this is, I'm really limited, in this diet, what I can do. And so he's studying specific proteins already that vary in some of these different breeds, and pinpointed this protein called MES that's kind of a key thing. But they're just figuring it out. But if you pick that as a topic, there'd be lots of examples. For example, you could get out into the web where humans are trying to make methionine restricted diets and eat them, hoping that would help them in their health, like that. Yes. Madison, what are you thinking about so far?
Thinking about using the research that I'm doing at my internship in mercury analysis and star feathers, to sort of assess changes, like, I don't know, whatever plasticity I can sort of pin down, or like environmental impact regarding mercury concentration. The cedar feathers have been like localized to Alaska, and this is sampled from 2000 to 2018. So seeing if there's like a way to pin down a connection between changes that we've seen and the changes we've seen in the birds and changes we've seen in mercury concentration.
Mm-hmm. Yeah. And so I think that's potentially great, because you're out in that internship, and so you're surrounded by some expertise, and it might end up being that you could, find out about how you could use the mercury specifically, but I think this kind of challenge is a good one to bring to an internship, because you're not there as an employee, so every once in a while you get a chance to say, well, I have some questions about, this stuff. And they recognize you, or you're an intern, you're getting trained, and they're willing to talk to you about broader topics. And so you should bring the concepts from this course to some of the experts. Like, I don't know if you've met Dave Evers or any of the kind of the leaders out there yet or not, but he's the owner of he runs the whole thing. Yeah. Yeah. But you can ask Tim Needham and say, I'm kind of working on this. And if they have ideas, because it might shoot off, like they might say, well, did you know loons have different, shaped bills depending on some environmental factor like that, that might, be a little bit shifted away from maybe the toxicity, maybe not, maybe they have that, But yeah, I think that's good, and in this writing assignment coming up, you can write about that, because it's about your, individual interests and how those fit in like that. Yep. Luke, are you thinking about stuff?
Yeah, I was trying for a while to kind of relate it to my own thesis, but I think it's kind of pretty distantly related, but maybe you have some ideas. But I think I'm leaning towards the jellyfish. I think it's a cool example of phenotypic plasticity due to the environment, and a crazy life history, lifespan topic as well.
Mm-hmm. Yeah, Luke was telling me, so probably everybody maybe has heard in just like general news reports, there are these jellyfish that are immortal, right? Now, how does that work? And at least in this one species that Luke described, the jellyfish is an adult jellyfish for a while, and then it can just revert to a juvenile stage and grow up again. And that's a mechanism, it may be tied into the mechanism of a kind of immortality. And so that would be good. One of the things that brings up a thing, right, where a person can get out to an area that is like, what do people know about jellyfish? Like that. You know, like collectively, people know a fair amount of cephalopods, but what do they know about jellyfish? And the answer is, not very much, so there's not very much there. So you might have to go to a totally different organism and say, I'm working on a lifespan, right? You know, and so I'm gonna look at these mechanisms and include that, right? And that's true kind of for everybody, is that you might need to go to just other species and say, you know, in a like a really extended background, but diving into the science of it, you know, where are we at with understanding lifespan and mechanism of lifespan like that. But yeah, I think those all sound like, you know, we're in a good shape so far. And again, I think, you know, I want to try to be helpful, and to do that, you know, we'll have to schedule some time outside of class. And so now I'm just going to scroll forward because we have a little bit more, because the next thing we're going to do a week from today is about symbiosis. And the book really focuses on the concept of microbiomes. And so there's an example that might be related to that Rob-Koza stuff, right? It might be that myothine restriction may include a real shift in the bacteria in the gut, you know, like that. And you know, you could look, you know, as part, like one section of your, you know, paper and project could be looking down that way. And what do we know about the role of the gut microbiome, like that? And in some cases you just feel, well, that we know a lot, and it's going to really be a big part of my final paper, you know. So, you know, kind of have a surprisingly shift toward something like that. And and so in chapter 3, the book goes into more of like examples and mechanisms, but especially wants to focus on the idea of symbiosis. And in doing that, it really wants to focus on the symbiosis between big plants and animals, especially animals, and microbes, like that. So that's a good one to think about and learn about, and as our next topic. And so we want to read that, and also get out into other areas. Maybe Bryce can bring more about it. You know, maybe you can learn more about methionine restriction in the gut microbiome or something like that, right? Because you're welcome to bring where you're at. Also, Juliet can bring, you know, maybe something, and we're still early, and maybe your topic will shift. But it's totally legit to have as one of the 5 things you send me, say, well, I've been working on something related to this, and so I can bring some of that, you know, like that to our discussion a week from today. So here's just one thing to talk about here, and actually maybe Ashton can bring something about this too, because in our digestive system, in our digestive tube, so our small intestine, there are patches of cells along it that are referred to as gut-associated lymphoid tissue. And that is where our immune system is developing. So it's a normal site of immune system development, and it includes paying attention to what the contents are in the digestive tube in order to encourage certain bacterial species to grow and to discourage others. So, you know, it's just a fact that, you know, our digestive tube is the external environment, and we're just hanging out with it more. You know, you can think about it like, you know, you're just holding your hand on there, and, you know, maybe you want to do something special with regard to that external environment. And so that's what it's about, and the relationship, the connection between our organismal systemic immune system and what's going on in the microbiome and other, you know, the biome environment in the digestive tube, like that. So our book wants to talk about that, talk about topics like probiotics and so forth, like that. Then this is a crazy example, but it's really been around for several years, and there were only like 2 or 3 lab research groups that were really pursuing this topic. And our book uses it as a big example for that microbiome concept, because it was a pioneer in that. And it's this story of this little squid that's like the size of your thumb, and it lives by Hawaii, and it's called the Hawaiian bob-tailed squid. And this is just a picture of what it's got that people are studying. And on its tummy, it has like a little pocket, like a kangaroo pocket. And in that tummy pocket, it collects bacteria from just the ocean water, and there's one particular species of bacteria that it's favoring based on a kind of mucus food that is making, the squid is making and putting it into that pocket, and that is going to encourage those bacteria to grow, and it's going to discourage others. Then every 24 hours, it burrows down in the sand, in the bottom of water, and it kind of pushes most of the bacteria out. Then it comes up and it starts over the next day, encouraging a certain species to grow. And this bacteria that grows is actually just very dilute in the ocean water. And it's not built to be thinking it's going to be in that pocket, so it's a really exceptional situation. And when the squid collects the bacteria start growing, and at a certain concentration they start to glow. And that's because for some reason, natural selection reason, this species of bacteria has a quorum sensing mechanism, and when the concentration of bacteria reaches a certain level, it starts to bioluminesce. And so that's shown in this drawing here too, that the pocket is referred to as a light organ, and it glows. And the best interpretation is that it's glowing, and it's making it harder for predators below to spot the bobtailed squid, like that. And people are skeptical about that explanation because you can see it's pretty active at night, and you're like, is that really what's going on? And people say, well, the sky is still really bright with the moon up there, and these predators are looking up, and they're expecting to see a shadow rather than a light source like that, And so, and I'm not keeping up with this literature, maybe that shifted something like that. But this is a really famous example because this is where that whole quorum sensing mechanism was first described. And then much of the story of how an organism encourages the microbiome, right? So the symbiosis is a two-way street with that gut-associated lymphoid tissue in us and other vertebrates, and that little light organ. So they revealed a lot of the principles and examples of how would an organism encourage a certain species of bacteria to grow in that setting like that. So you'll see that's an example, and to such a degree that our authors set that example and other symbiotic examples out into a separate chapter. And for all of us, it just brings up more examples of, whoa, I didn't think about that, but those symbiotes are always part of the environment, any of these relationships, right? And we've seen, some of our projects will relate to this. So that's what we're going to do. How we're doing for time, we're done. But there's also, in the environment, other topics like the uterine environment and this Dutch hunger winter, so we might get into some of those next week, but it'll especially actually be focused by the symbiotic relationships. Okay? All right, so we're going to stop. Like I said, I'm going to post this first, two-page, single-spaced writing assignment.